Then third one is Ascaris lumbricoides, which causes ascariasis. And fourth one is trichophyton ringworm disease, which is caused by trichophyton rubra. ಇದನ್ನು ಕೂಡ ಅದಲ್ಲ ಈ ಪ್ರಾಕ್ಟಿಕಲ್ ಹಾಕಿರ್ತೀನಿ. ಮೊದಲು ರೀಪ್ರೊಡಕ್ಷನ್ ಒಳಗಿನ ಪ್ರಾಕ್ಟಿಕಲ್ಸ್ ಮುಗಿಲಿ, ಆಮೇಲೆ ನೆಕ್ಸ್ಟ್ ಇದು ಹಾಕ್ತೀನಿ ಅಂತ. ಓಕೆ, ಸೊ the diseases that we have studied are: first one is common cold, second one is typhoid, third one is pneumonia, fourth one is ascariasis, fifth one is elephantiasis, sixth one is ringworm, seventh one is AIDS, and last one is cancer. And today let us study about the next part called as immunity. Followed, this immunity is IMMUNITY. So this immunity is the resistant capacity of an individual, resistant capacity or the capacity of an individual to resist a disease. Followed, the capacity of an individual to fight against the disease causing microorganisms. The resistant capacity of an individual to fight against the pathogens is called as immunity. And this immunity is due to presence of WBCs. As you know that these WBCs are the cells, are the corpuscles present in the blood. Followed, a person is having two types of immunity. One is called as, that is innate immunity, and second one is called as acquired immunity. Innate immunity and second one is called as acquired immunity. This innate immunity is immunity which is present from the birth of an organism, the birth of an individual. ಹುಟ್ಟಿದಾಗೆ ಇರುವಂತದ್ದು. ಆಯ್ತಾ? ಏನ್ ಬರೀಲಿಲ್ಲಮ್ಮ? What you have not written? Practical part. Practical part. Second practical. Okay, I'll send second practical as well as third practical today. You note down that one. Okay. So immunities are of two types. The first one is called as innate immunity. This innate immunity is a non-specific type of immunity. Non-specific ಅಂತಂದ್ರೇನು? ಸ್ಪೆಸಿಫಿಕ್ ಇರ್ಲಾರ್ದಂತದ್ದು. ಸ್ಪೆಸಿಫಿಕ್ ಇರ್ಲಾರ್ದಂತದಕ್ಕೆ ಏನಂತ ಕರಿತೀವಿ? We call it as non-specific. ಹಂಗಂದ್ರೆ ಏನು? ಯಾವುದೇ antigen center ಆಗಲಿ, the antibodies are being produced. ಆಯ್ತಾ? ಅಂತ immunityಗೆ ಏನಂತ ಕರಿತೀವಿ? We call it as non-specific type of immunity. Followed. So non-specific type of immunity is present in all, that is all the individual right from birth, and the response is same in all the individuals. ಎಲ್ಲಾರೊಳಗೆ ಆ same type of antigens produce ಆಗ್ತಾ ಇರ್ತಾವು, antibodies produce ಆಗ್ತಾ ಇರ್ತಾವು. Followed, and this non-specific type of immunity, that is innate immunity, consists of four types of barriers. It is having four different types of barriers which are divided into, that is physical barriers are there, physiological barriers are there, cytokine barriers are there, and fourth one are called as interferon barriers. Followed, or those are called as cellular barriers. So different types of barriers that are present in innate immunity. First one is called as innate immunity. Innate immunity. And the four different types of barriers are: first one is physical barrier, which includes skin and mucous membrane. Second one is a physiological barriers. Third one are called as cellular barriers. And fourth one are called as cytokine barriers. Cytokine barriers. So different types of barriers are there. Followed. So first one is called as, this first one is the physical barrier. This physical barrier includes the skin and mucous membrane. It includes the skin and mucous membrane. As you know that the skin, which is sensitive in nature, is the outer covering of the body. It covers the entire outer parts of the body. It is protective in nature. The skin, which is keratinized, which is outermost layer of our body, it prevents the entry of microorganisms. It prevents the entry of microorganisms. Apart from that, our skin produces two types of, that is, our skin consists of two types of glands such as sebaceous glands as well as sweat glands are there. These sebaceous glands, these are the oily glands. Followed, our skin appears to be shining in nature because of the sebaceous glands, which prevents the growth of microorganisms. It prevents the growth of microorganisms. The sweat glands produces this sweat. This sweat is acidic in nature, which that is destroys the outer membrane of the bacteria. So the fungal infection, the bacterial infection doesn't occur to a skin because of these two glands, sebaceous glands as well as the sweat glands. So the skin produces the first outermost layer which prevents the entry of microorganisms. Followed, and second one is the internal organs. Our internal organs such as digestive system, circulatory system, a reproductive system, excretory system, they are all lined by a mucous membrane. They are lined by a mucous membrane is there. It is gelatinous in nature. And that mucous membrane which is found lining the internal organs, they protect, this mucous membrane protects the innermost, there is a important systems of our body by engulfing or by trapping these microorganisms. Keratin is the outermost, that is the cells that are present in the outermost layer of the skin. Followed? So these, that is, it protects the entry of this mucus, it protects the entry of microorganisms inside our body. So these are the two physical barriers which prevents the entry of microorganisms or which prevents the entry of the pathogens in our body, which are included under physical barriers. Got the idea now? Second one is physiological barriers. Physiological barriers. Istella skin idranu kuda, sometimes what happens during eating, some of these microorganisms may enter our mouth through the food. Followed, at that time in our mouth, that is mouth is called as in that is a mouth, the open part of the mouth is called as oral cavity or buccal cavity. This oral cavity or buccal cavity consists of upper and lower jaw, that is jaws are there, and this upper and lower jaws are beset with different types of teeth called as heterodont, and the four different types of teeth are incisors, canines, premolars, and molars, and the dental formula is written as 2 1 2 3 by 2 1 2 3, isn't it? So these teeth help in chewing the food material. They help in grinding the food material. After grinding the food, the tongue, which doesn't have bones, which consist of only the taste buds, this tongue, it rolls the food into a round mass, isn't it? And that round mass is called as bolus. It is called as bolus. And this bolus has to be swallowed. After swallowing, it enters into the pharyngeal region. From the pharynx, it enters into the esophagus. From the esophagus, it reaches the stomach, isn't it? So while swallowing, this bolus, what is receives? It receives saliva. It receives saliva. And this saliva is produced by three different types of salivary glands. A pair of parotid glands, a pair of sublingual glands, and a pair of submaxillary glands. Followed? All these glands, they produce saliva. And the secretion of saliva is initiated by the taste of the food. Tastier the food, more is the secretion of saliva. It is secreted during the imagination of the food. ಬಹಳ ಹಸಿವಾಗಿರ್ತದ, ಹಂಗೆ ಕಾಲೇಜ್ ಒಳಗೆ ಕೂತಿರ್ತೀವಿ, ಮನ್ಯಾಗ ಇವತ್ತೇನೋ ಸ್ವೀಟ್ ಮಾಡ್ಯಾರ ಅಂತಂದ್ರೆ ಅವ ಬಾಯಿ ಒಳಗೆ ನೀರ್ ಬರ್ಲಿಕ್ಕೆ ಶುರು ಆಗ್ತದೆ. Followed, that is imagination of the food also, the secretion of saliva increases. By sight of the food, ಯಾರಾದ್ರೂ ಹಿಂಗ ಫುಡ್ ತಗೊಂಡ್ ಹೋದ್ರು ಅಂತಂದ್ರ ಅವ ಬಾಯಿ ಒಳಗೆ ನೀರ್ ಬರ್ಲಿಕ್ಕೆ ಶುರು ಆಗ್ತದೆ. So this saliva so produced, it lubricates the food. Apart from that, this saliva consists of there is one enzyme named lysozyme. This lysozyme is having antiviral and antibacterial characteristics. Is having antiviral and antibacterial characteristics, which kills the viruses and bacteria, the microorganisms that are present in the food material, and allows only the that is food free from microorganism to enter into the pharynx, and from the pharynx it enters into the esophagus. From the esophagus it reaches the stomach. ಅಕಸ್ಮಾತ್ ಆ ಲೈಸೋಸೈಮ್ಲಿಂದು ಕೂಡ ಈ ಬ್ಯಾಕ್ಟೀರಿಯಾಸ್ ವೈರಸಸ್ ಅಥವಾ ಮೈಕ್ರೋ ಆರ್ಗಾನಿಸಮ್ಸ್ ಕಿಲ್ ಆಗ್ಲಿಲ್ಲ ಅಂತಂದ್ರ, at that time the food, the bolus, along with the bolus, some of the microorganisms may reach our stomach region. In the stomach, you know that stomach is a J-shaped bag-like structure which is divided into 4 parts. That is pyloric stomach, cardiac stomach, body, and fundic, isn't it? So this stomach, which is J-shaped, is internally lined over by gastric mucosa. It is lined by gastric mucosa, and this gastric mucosa consists of gastric glands. And these gastric glands, they produce gastric juice. They produce gastric juice. Along with this gastric juice, HCl is being secreted. So as soon as this bolus touches the gastric mucosa, that is the inner lining of the stomach, what happens? This gastric mucosa produces one hormone named gastrin. It produces one hormone named gastrin. And this gastrin stimulates the gastric glands. The gastric glands consist of 2 cells, peptic cells and oxytonic cells are there, and these cells, they go on producing the gastric juice as well as HCl. No doubt this gastric juice consists of different types of digestive enzymes such as pepsin, renin, and gastric lipase are there, which acts on the food material. They act on protein and fatty part of the food material. And the function of this HCl is, what it does, this HCl, which is acidic in nature, hydrochloric acid, this HCl kills the microorganisms that are present in the food. It paralyzes the microorganisms that are present in the food. It deactivates the microorganisms that are present in the food, and dissolves the that is insoluble minerals, activates pepsin and renin. Apart from that, what it does, this HCl, it loosens the fibrous components that are present in the food material. So almost all the microorganisms are killed by this HCl, which is produced in the stomach region. Followed, and apart from that, from here the food that is gets converted into a semi-solid mass called as chyme. The chyme enters into the small intestine. Complete digestion occurs in the duodenum. The digested food is absorbed in the jejunum and ileum, and later the undigested food enters into the large intestine, and the process continues. We are concerned with the immunity part. The physiological barrier include, first one is saliva containing lysozyme, and second one is gastric juice containing HCl. Apart from that, the tear glands, called as lacrimal glands. Lacrimal glands, these lacrimal glands also contain certain antibacterial properties are there. Whenever something falls, whenever some particle enters our eyes, immediately what happens? It starts watering. Followed, because the lacrimal glands, they go on producing the tears. Followed, along with the tears, what happens? That waste also comes out. So crying is healthier. ಯಾವಾಗಲೂ ಅಳ್ತಾ ಇರಬೇಕು ಅಂತ ಹೇಳ್ತಾರಲ್ಲ, ಯಾಕಂತಂದ್ರ it keeps away the microorganisms. Followed, the next one is, apart from that, the wax which is produced in our ears. Wax which is produced in our ears, ಗೂಗಣ ಅಂತ ಹೇಳ್ತೀವಲ್ಲ, ಅದನ್ನು ಏನ್ ಮಾಡ್ತಾ ಇರ್ತದ ಅಂತಾರೆ? It entangles the microorganism that enters our ears. Followed, so these are the 4 different types of physiological barriers which prevents the entry of microorganisms from our body. Followed, the next one is the cellular barriers. Cellular barriers includes the cells. The cellular barriers includes the WBCs, white blood corpuscles. As you know that these white blood corpuscles are without the pigment. Followed, and these are amoeboid in shape. These cells are shapeless. They are amoeboid in shape, which moves from white wander from one place to another place and are nucleated. Followed, they are having a lobed nucleus is there. They are having lobed nucleus. So different types of cells, that is such as these WBCs are called as leukocytes, called as polymorphonuclear leukocytes, PMNL. These are polymorphonuclear leukocytes. Polymorphonuclear leukocytes, PMNL, and different types of neutrophils, then macrophages. These are different types of WBCs only. Followed, polymorphonuclear leukocytes, then there is monocytes, natural killer cells, macrophages. These are all. Then these are all phagocytic in nature. Different types of WBCs even. ಆಯ್ತಾ, ಯಾವುದು ಒಂದೇದು PMNL ಅಂತಾರ, ಪಾಲಿಮಾರ್ಫೋನ್ಯೂಕ್ಲಿಯರ್ ಲ್ಯೂಕೋಸೈಟ್ಸ್, ನ್ಯೂಟ್ರೋಫಿಲ್ಸ್ ಅಂತ ಇರ್ತಾವ, ಲೋಬ್ಡ್ ನ್ಯೂಕ್ಲಿಯಸ್ ಇದ್ದಂತಾವು. ಆಮೇಲೆ ಮ್ಯಾಕ್ರೋಫೇಸಸ್ ಇರ್ತಾವು, ವಿಚ್ ಆರ್ ಅಮೀಬಾಯ್ಡ್ ಇನ್ ಶೇಪ್. ಮೊನೋಸೈಟ್ಸ್ ಅಂತ ಇರ್ತಾವು. ನ್ಯಾಚುರಲ್ ಕಿಲ್ಲರ್ ಸೆಲ್ಸ್ ಅಂತ ಇರ್ತಾವು, NK cells, ನ್ಯಾಚುರಲ್ NK cells ಅಂತ ಹೇಳಿ. All these are different types of WBCs, and these different types of WBCs, they wander from one place to another place inside our body and engulf, they go on swallowing. They are phagocytic in nature, which swallows the solid objects, the disease causing microorganisms, and defend our body against the cause of the disease. Data. So these are different types of barriers, cellular barriers. And the fourth type of barriers are called as cytokine barriers. These are called as cytokine barriers. Followed. So these cytokine barriers, that is, whenever, sometimes, apart from this physical barrier, physiological barrier, cellular barrier, some of these viruses, they escape and infect our cell. For example, HIV is there. It infects the helper T cell, that is the macrophages are being infected. At that time, what happens? As I told you earlier, that is, it leaves the RNA. This RNA produces a DNA. This process is called as a reverse transcription, which occurs in the presence of an enzyme named reverse transcriptase. This DNA combines with, or it combines with the host DNA. And this host DNA as well as viral DNA, they go on producing, that is the viral particles. So it acts as a machine, and these viruses are being produced. So the cell, virus infected cell, that is produces certain proteins called as interferons. It produces certain protein. Yaru? Barre. So Ramai, so it is infected by, that is, when a cell is infected, when number of viruses are produced inside this cell, at that time this infected cell produces certain antiviral proteins. It produces certain antiviral proteins, and these antiviral proteins are called as interferons. These are called as interferons, which, that is, protects the non-infected cell from infection of the virus. ಈ cell ಅನ್ ಕೈಗೆ ಇನ್ಫೆಕ್ಟ್ ಆಗ್ಬಿಟ್ಟಿರ್ತದ, ಬಾಜು ಸೆಲ್ ಇನ್ಫೆಕ್ಷನ್ ಆಗಿರೋದಿಲ್ಲ. ಅದಕ್ಕೆ ಈ infected cell, what it goes on producing the antiviral proteins. Antiviral proteins ಏನಂತ ಕರೀತಾರ, those are called as interferons. Interferons ಏನ್ ಮಾಡ್ತಾವ ಅಂತಂದ್ರ ಈ ನಾನ್ ಇನ್ಫೆಕ್ಟೆಡ್ ಸೆಲ್ಸ್ ಅನ್ನ ರಕ್ಷಣೆ ಮಾಡ್ತಾ ಇರ್ತಾವ. They protect the cells that are not infected by the virus. They prevent the infection of the virus to the cell, normal healthy cell. And such type of barriers are called as cytokine barriers. Got the idea now? This innate immunity is a non-specific type of immunity wherein the specific, that is a response in all of the individual is similar to different types of antigens. Followed? So it consists of 4 different types of barriers. The first barrier is the physical barrier, which includes skin and mucous membrane, and second one is physiological barrier. The third one are called as cellular barriers, and fourth one is called as cytokine barriers. The second type of immunity is called as acquired immunity. Second type barrier is called as acquired immunity. So what is this acquired immunity? This acquired immunity is the immunity which is acquired after the birth of an individual, or which is acquired during the lifetime of an individual. ಹೊರಗಡೆಯಿಂದ ಬಂದಂತ ಆ ಇಮ್ಯುನಿಟಿಗೆ ನಾವೇನಂತ ಕರಿತೀವಿ, ವಿ ಕಾಲ್ ಇಟ್ ಆಸ್ ಅಕ್ವೈರ್ಡ್ ಇಮ್ಯುನಿಟಿ. ಆಮೇಲೆ ನಾವು ಹುಟ್ಟಿದ ತಕ್ಷಣ ಆ ಹುಟ್ಟಿದ ಮೇಲ್ಗಡೆ ಬಂದಂತ ಇಮ್ಯುನಿಟಿ, ಇಟ್ ಇಸ್ ಸ್ಪೆಸಿಫಿಕ್ ಟೈಪ್ ಆಫ್ ಇಮ್ಯುನಿಟಿ. Specific type of immunity antara when certain type of antigens enters our body, a particular antibodies produce ಆಗ್ತವೆ. ವೈರಸ್ ಆಂಟಿಜನ್ ಇತ್ತು ಅಂತಾರ ಆ ವೈರಸ್ ಅನ್ನ ಕಿಲ್ ಮಾಡುವಂತ ಆಂಟಿಬಾಡೀಸ್ ಪ್ರೊಡ್ಯೂಸ್ ಆಗ್ತವೆ. ಬ್ಯಾಕ್ಟೀರಿಯಲ್ ಆಂಟಿಜನ್ ಇತ್ತು ಅಂತಾರ ಬ್ಯಾಕ್ಟೀರಿಯಾ ಕಿಲ್ ಮಾಡುವಂತವೇ ಆಂಟಿಬಾಡೀಸ್ ಪ್ರೊಡ್ಯೂಸ್ ಆಗ್ತಾ ಇರ್ತಾವು. ಆಯ್ತಾ? ಸ್ಪೆಸಿಫಿಕ್ ಟೈಪ್ ಆಫ್ ಇಮ್ಯುನಿಟಿ ಇಸ್ ವೆರಿ ಸ್ಪೆಸಿಫಿಕ್, which is based on antigen and antibody reaction. Followed. So what are these antigens? What are antigens? What are antigens? Specific immunity adala, it is based on this antigen antibody reactions. Antigens andre enu? Is anybody? These are the foreign substances. These are the foreign substances which induces our immune system for the production of antibodies. Notebook ada ನಿಮ್ಮ ಹತ್ರ ಎಲ್ಲರ ಕಡೆ? Notebook ada? You are having notebook with you?
Yes, ma'am.
Okay.
Yes, ma'am.
Antigen ಬರ್ಕೊಡ್ರಿ ಅಂದ್ರೆ ಗೊತ್ತಾಗ್ತದೆ ಇದು. Antigen. These are the foreign substances. Foreign substances. ಹೊರಗಡೆಯಿಂದ ಏನ್ ಒಳಗಡೆ ಹೋಗ್ತಾವಲ್ಲ ಅವಕ್ಕೆಲ್ಲ ಆಂಟಿಜನ್ಸ್ ಅಂತೀವಿ. ಆ ಫಾರಿನ್ ಸಬ್ಸ್ಟೆನ್ಸ್ ಹೆಂಗಿರ್ಬೋದು, ವೈರಸ್ ಇರ್ಬೋದು, ಬ್ಯಾಕ್ಟೀರಿಯಾ ಇರ್ಬೋದು, ಒಮ್ಮೊಮ್ಮೆ ವೀಟ್ ಇರ್ಬೋದು, ಗೋಧಿ ತಿಂದ್ರೆ ಒಬ್ರೊಬ್ರಿಗೆ ಅಲರ್ಜಿ ಆಗ್ತಾ ಇರ್ತದ, ಒಮ್ಮೊಮ್ಮೆ ಮಿಲ್ಕ್ ಇರ್ಬೋದು, ಚಾಕ್ಲೇಟ್ ಇರ್ಬೋದು, ಐಸ್ ಕ್ರೀಮ್ ಇರ್ಬೋದು, ಯಾವ್ದಾದ್ರೂ ಇರ್ಬೋದು. These are the foreign substances which induces 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 our immune system for the production of antibodies. 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 ಬರದ್ರಿ? Next, what are antibodies? Antibodies andre.
Antibodies are the foreign substances.
Antibodies. Antigens are the foreign
Antigens, ma'am.
Yes.
Proteins.
Substances. So antibodies.
Antibodies are the proteins.
Antibodies are the proteins which are produced in response to the antigens. Idu one barkodri. Antibodies. antibodies. These are the proteins which are produced in response to the antigens. Rita? So when certain antigens they enter our body, at that time our body responds to it and produces antibodies. ಅರ್ಥ ಆಗ್ಲಿಕತ್ತದ? ಸೊ ನಮ್ಮ ಬಾಡಿ ಒಳಗೆ ಪ್ರೊಡ್ಯೂಸ್ ಆದಂತವೇನು ಅಂತಾರೆ antibodies. ಈ ಹೊರಗಡೆ ಎಂಟರ್ ಆದಂತದ್ದೇನು antigen. ಆ antigen ಪ್ರೋಟೀನ್ ಇರ್ಬೋದು, ಕಾರ್ಬೋಹೈಡ್ರೇಟ್ ಇರ್ಬೋದು, ಫ್ಯಾಟ್ ಇರ್ಬೋದು. ಹೌದಿಲ್ಲ? So this is a foreign substance which induces our immune system for the production of antibodies. So antibodies are the proteins which are produced in response to the antigens. Followed? So this antibody, where these antibodies are present, these antibodies are present in our body. Followed? They are present in our body. And these antibody, when we observe the structure of this antibody, this antibody is made up of, will you draw the diagram of this antibody? Right heading is structure of antibody. Structure of antibody. This antibody it is a protein, means it is made up of polypeptide chain. 2 polypeptide chains are there, and these 2 polypeptide chains are arranged in the form of Y-shaped structure. They are arranged in the form of Y-shaped structure. Well, this is the structure of an antibody, and each antibody consists of, I'll send you the diagram of this one. Each antibody, it is made up of 2 polypeptide chains are there. Followed? It has 2 polypeptide chains, and these 2 polypeptide chains are, one is called as, there is a heavy chain, and another one is called as a light chain. An antibody molecule is made up of 4 polypeptide chain arranged in Y-shaped manner. It is arranged in a Y-shaped manner. There are 2 long chain called as heavy chain. It consists of 2 long chain called as heavy chain and 2 short chain called as a light chain. Followed. So it is represented as H2L2. Is represented as H2L2. H2 means two heavy chains and two light chains are there. Followed. And these two, that is all the four chains, are held together by disulfide bonds are there. They are held in contact with one another by disulfide bond. A polypeptide chain that is consists of two regions, namely one is called as variable region and second one is that is constant region. It has two regions, one is called as variable region and second one is called as constant region. The variable region is the antigen binding site. This is the variable region called as antigen binding site, whereas the second one is called as the constant region, which remains the same for all types of antibodies. It remains the same for all types of antibodies. The different types of antibodies that are present in our body are IgA. I means immunoglobulins. IgA. IgA, IgE, IgG, IgM, IgD. Five different types of antibodies are there. IgA, IgE, IgG, IgD, IgM. Followed. So this is the structure of antibody. Got the idea now? These antibodies, an antibody molecule consists of four polypeptide chains which are arranged in the form of Y-shaped structure. Two chains are heavy chains and two are light chains. Heavy chains are made up of about 400 amino acid. Light chains are made up of 200 amino acid. ಆಯ್ತಾ? ಈ ಹೆವಿ ಚೈನ್ ಮತ್ತೆ ಲೈಟ್ ಚೈನ್ ಎರಡು ಅಮೈನೋ ಆಸಿಡ್ ಇಂದ ಮಾಡಲ್ಪಟ್ಟಿರ್ತಾವಲ್ಲ, ದೇ ಆರ್ ರಿಪ್ರೆಸೆಂಟೆಡ್ ಆಸ್ H2L2. Followed. See, all the four chains are held together by disulfide bond. And the polypeptide chain consists of two regions. One is called as variable region and second one is called as constant region. This variable region consists of antigen binding site to which the antigen comes and attach, and second one is called as a constant region which remains same for all types of the antibodies. Followed. And the different types of antibodies that are present in our body are IgA, IgD, IgE, IgG, IgM. Followed. And next one, this acquired immunity. Got the idea now? Acquired immunity is carried out by the specific type of lymphocytes. Specific type of lymphocytes, specific type of WBCs are there. The first one is called as B lymphocyte. Second one, first one is called as B lymphocyte and second one is called as T lymphocyte. B lymphocytes and second one are called as T lymphocytes. T lymphocyte. B lymphocyte and T lymphocyte. So how this B lymphocyte and T lymphocyte, they play active role in defending our body. How they are, that is, they kill, or how they are involved in creating the resistant capacity in our body. How they fight against disease causing microorganisms. See, these B lymphocytes are the types of WBCs which are produced in the bone marrow. Followed. These are produced in the bone marrow. They differentiate in the bone marrow as well as they mature in the bone marrow itself. So these are called as B lymphocytes. B means which gets differentiated and matures in the bone marrow only. And these B lymphocyte acts indirectly against the antigens, against the disease causing microorganisms, against the pathogens by producing the antibodies. Followed. They indirectly, they immobilizes or they indirectly help in killing the microorganisms, and this type of immunity is called as, that is, antibody mediated immunity, or it is otherwise called as humoral immunity. It is called as antibody mediated immunity, or it is otherwise called as humoral immunity. Wait for a minute. Yes, antibody mediated immunity. Mediated immunity, or it is called as humoral immunity. Humoral immunity. The immunity which is produced by this B lymphocyte is called as antibody mediated immunity, or is otherwise called as humoral immunity. HUMORAL. Humoral immunity. Followed. So these lymphocytes, they act indirectly, followed by producing the antigens, by producing the antibodies. The antibodies are carried by the body fluid by like blood and lymph. Followed, they go on producing. When an infection occurs in the hand region, at that time, immediately the message is transmitted to the antibodies. At that time, this B lymphocyte, they get activated. Followed, and these B lymphocyte, they produce a specific type of antibodies. And these antibodies, what happens? These antibodies, they are transmitted to the site of infection along with the blood, that is the body fluid. Body fluid ಯಾವ್ಯಾವು ನಮ್ಮಲ್ಲಿ blood ಮತ್ತೆ lymph, and reacts in various ways to destroy the specific antigens. Followed, and when these antigens enters into the tissues or the cells, they multiply at a faster rate and produces a clone of plasma cells and memory cells. The plasma cells produces the antibody which neutralizes the antigens, and such type of immunity is called as humoral immunity. ಗೊತ್ತಾಯ್ತು ಬಿ lymphocytes ಬಗ್ಗೆ. The lymphocytes which are produced in the bone marrow and matures there only. These B lymphocytes indirectly, or that is they immobilize or they kill the pathogens indirectly by producing the antibodies, and such type these antibodies so produced are transmitted to the site of action along with the body fluids such as blood and lymph and neutralizes the effect. Followed, and such type of immunity is called as antibody mediated immunity, or is otherwise called as humoral immunity. The second type of immunity is called as, the second type of lymphocytes are called as T lymphocytes. T lymphocytes. Followed, and these T lymphocytes, no doubt they are produced in the bone marrow. From the bone marrow, they migrate to the thymus region. Thymus glands are present just near to the heart region. Our thymus glands ನಲ್ಲಿ ಏನಾಗ್ತಾವ ಅಂತಾರೆ, they get activated and they mature there. Followed, ಎಲ್ಲಿ ಪ್ರೊಡ್ಯೂಸ್ ಆಗಿರ್ತಾವ, ಬೋನ್ ಮ್ಯಾರೋ ಒಳಗೆ ಪ್ರೊಡ್ಯೂಸ್ ಆದಂತ lymphocytes ಅವಲ್ಲ, these lymphocytes from the bone marrow, they migrate to the thymus gland and matures there. So such type of lymphocytes are called as a T lymphocytes. Followed, and these T lymphocytes, what they do, they act directly against the antigens by attacking themselves to the antigen bearing cells. They directly go on. ಇವೆಲ್ಲ B lymphocyte indirect ಆಗಿ antibodies produce ಮಾಡಿ ಅವನ್ನ ಅಟ್ಯಾಕ್ ಮಾಡ್ತಾ ಇರ್ತಾವ. ಇವು ಹಂಗಲ್ಲ, ಅಲ್ಲೇ ಹೋಗಿ ಆ ಸೆಲ್ಸ್ ಅಟ್ಯಾಕ್ ಮಾಡಿ ಅವನ್ನ ಕಿಲ್ ಮಾಡ್ಬರ್ತಾರೆ. ಆಯ್ತಾ? ಯಾವ T lymphocytes. These T lymphocytes act directly against the antigen by attacking themselves to the antigen bearing cell. And such type of immunity is called as cell mediated immunity. Anta immunity ಏನಂತ ಕರೀತಾರ? It is called as a cell mediated immunity. And this cell mediated immunity is brought about by different types of cells. Actually, these T lymphocyte, they produce, there are different, that is these cells are differentiated into different types to perform different functions. Followed, T lymphocyte ನಲ್ಲಿ ಬೇರೆ 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 ತರ cells ಇರ್ತಾವು. These are called as killer T cells, helper T cells, suppressor T cells, memory T cells. ಆಯ್ತಾ? ಟಿ ಸೆಲ್ಸ್ ನಲ್ಲಿ ಮತ್ತೆ 4 ತರ ಸೆಲ್ಸ್ ಇರ್ತಾವೆ. ಒನ್ ಆರ್ ಕಾಲ್ಡ್ ಆಸ್ ಕಿಲ್ಲರ್ ಟಿ ಸೆಲ್ಸ್. These killer T cells, they migrate to the site of infection and attaches to the invading microbe or antigen and secretes the toxic substances that destroy the microbes directly. ಕಿಲ್ಲರ್ ಟಿ ಸೆಲ್ಸ್ ಅವಲ್ಲ, ಕಿಲ್ಲರ್ ಟಿ ಸೆಲ್ಸ್ ಏನಾಗ್ತಾವ ಅಂತಂದ್ರ ಪ್ರೊಡ್ಯೂಸ್ ಆದ್ವು ಅಂತಂದ್ರ ಎಲ್ಲಿ ಇನ್ಫೆಕ್ಷನ್ ಆಗಿರ್ತಾವ ಅಲ್ಲಿಗೆ ಬಂದ್ಬಿಡ್ತಾವ ಡೈರೆಕ್ಟ್ ಆಗಿ. ಆ ಡೈರೆಕ್ಟ್ ಬಂದು ಆ ಸೆಲ್ ಇನ್ಫೆಕ್ಟ್ ಆಗಿರ್ತದಲ್ಲ, ಆ ಸೆಲ್ ಒಳಗೆ ಏನ್ ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡ್ತಾವಂತಾರ, ಆ ಆಂಟಿಬಾಡೀಸ್ ಅನ್ನ ಪ್ರೊಡ್ಯೂಸ್ ಮಾಡಿ ಆ ಪೂರ್ತಿ ಪ್ಯಾಥೋಜನ್ ಅನ್ನ ಡಿಸ್ಟ್ರಾಯ್ ಮಾಡಿ ಕಲಾಸ್ ಮಾಡಿ ವಾಪಸ್ ಅಂತ ಸೆಲ್ಸ್ ನಾವೇನಂತ ಕರಿತೀವಿ, ವಿ ಕಾಲ್ ದೆಮ್ ಆಸ್ ಕಿಲ್ಲರ್ ಟಿ ಸೆಲ್ಸ್. ಹೋಗಿ ಅವ್ನ್ ಕಲಾಸ್ ಮಾಡಿ ವಾಪಸ್ ಬಂದ್ಬಿಡುತ್ತೆ. ಸೆಕೆಂಡ್ ಒನ್ ಆರ್ ಕಾಲ್ಡ್ ಆಸ್ ಹೆಲ್ಪರ್ ಟಿ ಸೆಲ್ಸ್. ಹೆಲ್ಪರ್ ಟಿ ಸೆಲ್ಸ್ ಏನ್ ಮಾಡ್ತಾವ್, ದೀಸ್ ಸೆಲ್ಸ್ ಸ್ಟಿಮ್ಯುಲೇಟ್ಸ್ ದಿ ಬಿ ಸೆಲ್ಸ್ ಟು ಪ್ರೊಡ್ಯೂಸ್ ಆಂಟಿಬಾಡೀಸ್. ಬಿ ಸೆಲ್ಸ್ ಅವಲ್ಲ, ಈ ಬಿ ಲಿಂಫೋಸೈಟ್ಸ್ ಏನ್ ಮಾಡ್ತಾವ ಅಂತಂದ್ರ ಸ್ಟಿಮ್ಯುಲೇಟ್ ಮಾಡ್ತಾ ಇರ್ತಾವ ಆಂಟಿಬಾಡೀಸ್. ಯಾಕಂತಂದ್ರ ಈಗ ಟಿ ಸೆಲ್ಸ್ ಸ್ಟಿಮ್ಯುಲೇಟ್ ಆಗಿರ್ತಾವ. ಟಿ ಸೆಲ್ಸ್ ಸ್ಟಿಮ್ಯುಲೇಟ್ ಆದಾಗ ಬಿ ಸೆಲ್ಸ್ ಏನು ಫಾರ್ಮೇಶನ್ ಮಾಡ್ತಾ ಇರೋದಿಲ್ಲ. ಅವಾಗ ದಿ ಸೆಲ್ಸ್ ದಿ ಗೋ ಆನ್ ಸ್ಟಿಮ್ಯುಲೇಟಿಂಗ್ ದಿ ಬಿ ಸೆಲ್ಸ್ ಟು ಪ್ರೊಡ್ಯೂಸ್ ದಿ ಆಂಟಿಬಾಡೀಸ್ ವಿಚ್ ಇಂಟರಾಕ್ಟ್ ವಿತ್ ದಿ ಆಂಟಿಜನ್ಸ್ ಆಫ್ ದಿ ಮೈಕ್ರೋಬ್ಸ್. Third one are called as suppressor T cells. These cells suppresses the immune system after the antigen or the microbe is destroyed. ಈಗ ಆಂಟಿಜನ್ ಮೈಕ್ರೋಬ್ ಡಿಸ್ಟ್ರಾಯ್ ಹೋಯ್ತು ಅಂತಂದ್ರೆ ಅವಾಗ ಏನ್ ಪ್ರೊಡ್ಯೂಸ್ ಆಗಬಾರ್ದು, ಕಿಲ್ಲರ್ ಟಿ ಸೆಲ್ಸ್ ಪ್ರೊಡ್ಯೂಸ್ ಆಗಬಾರ್ದು, ಹೆಲ್ಪರ್ ಟಿ ಸೆಲ್ಸ್ ಪ್ರೊಡ್ಯೂಸ್ ಆಗಬಾರ್ದು. ಅದಕ್ಕೆ ಅವ್ನು ಸಪ್ರೆಸ್ ಮಾಡ್ಬೇಕಲ್ಲ, ಅವ್ನು ಇನ್ಆಕ್ಟಿವ್ ಮಾಡ್ಬೇಕಾದ್ರ ಯಾವ ಸೆಲ್ಸ್ ಪ್ರೊಡ್ಯೂಸ್ ಆಗ್ತಾವು, ಸಪ್ರೆಸರ್ ಟಿ ಸೆಲ್ಸ್ ಆರ್ ಬಿಂಗ್ ಪ್ರೊಡ್ಯೂಸ್ಡ್. Followed, and the fourth type of cells are called as memory T cells. ಮೆಮರಿ ಟಿ ಸೆಲ್ಸ್ ಹೆಂಗ ಅಂತಾರ, ಮೆಮರೈಸ್ ಮಾಡ್ಕೊಂಡಿರ್ತಾವ. ಇನ್ನೊಮ್ಮೆ ಅದೇ ಸೇಮ್ ಪ್ಯಾಥೋಜನ್ ಬಂದು ಇನ್ಫೆಕ್ಟ್ ಮಾಡ್ತು ಅಂತಾರ ಫಾಸ್ಟ್ ಆಗಿ ರಿಕವರಿ ಆಗ್ಬಿಡ್ತದೆ. ಆಯ್ತಾ? ಸೊ ದೀಸ್ ಸೆಲ್ಸ್ ಆರ್ pre-programmed to recognize and respond very quickly against an antigen that may appear in the future. Data. So these are the 4 different types of T cells. The first one are called as killer T cells. Second one are called as helper T cells. The third one are called as suppressor T cells, and fourth one are called as memory T cells. Followed. So this is all about the acquired immunity. Followed, and because of this T cells only, rejection of organ transplantation. That is, you might have seen an ad in the TV or newspaper that somebody is in the need of kidneys as the kidneys have failed to function. So at that time, there are many donors who can donate their kidney. ಈ ನಾವು ಕಿಡ್ನಿ ಡೊನೇಟ್ ಮಾಡ್ತೀವಿ ಅಂತಂದ್ರೆ ಇಮಿಡಿಯೇಟ್ಲಿ ತಗೊಳಿಕ್ ಬರೋದಿಲ್ಲ. ಯಾಕಂತಂದ್ರ ಟಿಶ್ಯೂ ಫಿಟ್ ಮ್ಯಾಚ್ ಆಗ್ಬೇಕು, ಬ್ಲಡ್ ಗ್ರೂಪ್ ಮ್ಯಾಚ್ ಆಗುದು, ಆಮೇಲೆ ಪಾಸಿಟಿವ್ ನೆಗೆಟಿವ್ ಮ್ಯಾಚ್ ಆಗುದು, ಅದೆಲ್ಲ ನಡೀತದ. ಬಟ್ ಏನ್ ಮ್ಯಾಚ್ ಆಗ್ಬೇಕು ಅಂದ್ರೆ ಟಿಶ್ಯೂ ಫ್ಲೂಯಿಡ್. ಆ ಟಿಶ್ಯೂ ಫ್ಲೂಯಿಡ್ ನಲ್ಲಿ ಎಷ್ಟೆಷ್ಟು ಕಂಟೆಂಟ್ ಇರ್ತಲ್ಲ ಅದೆಲ್ಲ ಮ್ಯಾಚ್ ಆಗ್ಬೇಕಾಗ್ತದೆ. ಇವರು ಹಾಕಿಸ್ಕೊಂಡ್ರಂತೂ ಕೂಡ, ಈ ಟ್ರಾನ್ಸ್ಪ್ಲಾಂಟೇಶನ್ ಆದ್ರೂ ಕೂಡ ಅವರ್ ಬಾಡಿ ಥಿಂಕ್ಸ್ ದಟ್ ಇಟ್ ಈಸ್ ದಿ ಫಾರಿನ್ ಸಬ್ಸ್ಟೆನ್ಸ್ ಅಂತೆ. ಫಾರಿನ್ ಸಬ್ಸ್ಟೆನ್ಸ್ ಅಂತಂದ್ರೆ ಏನು, ಆಂಟಿಜನ್ಸ್ ಅಂತ ಥಿಂಕ್ ಮಾಡ್ತಾ ಇರ್ತಾರೆ. ಆಂಟಿಜನ್ಸ್ ಅಂತಂದ್ರೆ ಏನ್ ಪ್ರೊಡ್ಯೂಸ್ ಆಗ್ತಾ ಇರ್ತಾವ ನಮ್ಮ ಬಾಡಿನಲ್ಲಿ, ಆಂಟಿಬಾಡೀಸ್ ಆರ್ ಪ್ರೊಡ್ಯೂಸ್. ಅವ ರಿಜೆಕ್ಷನ್ ಮಾಡ್ತಾ ಇರ್ತಾರೆ. ಆ ರಿಜೆಕ್ಷನ್ ಮಾಡ್ತಾ ಇರ್ತಲ್ಲ, ಅದಕ್ಕೆ ಏನ್ ಮಾಡ್ತಾರ ಅಂತಂದ್ರ ಆಂಟಿಬಾಡೀಸ್ ಆಂಟಿಬಾಡಿ ವ್ಯಾಕ್ಸಿನ್ಸ್ ಅವ ಅವನವ್ರೊಳಗೆ ಇಂಜೆಕ್ಟ್ ಮಾಡ್ತಾ ಇರ್ತಾರೆ. ಅಂದ್ರೆ ಆಂಟಿಬಾಡೀಸ್ ಪ್ರೊಡಕ್ಷನ್ ಕಡಿಮೆ ಆಗ್ಬೇಕಂತ. ಅಂದ್ರೆ ಇದು ನಮ್ಮ ಬಾಡಿಗೆ ಅಡ್ಜಸ್ಟ್ ಆಗ್ಬೇಕಾದ್ರೆ ಅಲ್ಲಿ ಏನ್ ಪ್ರೊಡ್ಯೂಸ್ ಆಗಬಾರ್ದು, ಆಂಟಿಬಾಡೀಸ್. Followed. So rejection of organ transplant like heart, kidney, eyes, liver are due to T lymphocytes. Tissue matching, blood group matching are essential for organ transplantation. Even after tissue typing, human suppressant is required before and after transplantation. Suppressant and ಹೇಳಿ vaccines are there. The immune system is able to differentiate self and non-self, and the cell-mediated immune response are responsible for organ injection. Followed. Then next one is about the active immunity and passive immunity. Now, what is difference between T lymphocytes and B lymphocytes? T lymphocytes and B lymphocytes. So these T cells, we call them as WBCs. T lymphocytes are produced in the bone marrow and matured in the bone marrow only. Whereas T lymphocytes, they are produced in the bone marrow, From the bone marrow, they migrate to the thymus region where they undergo differentiation as well as maturation. And these B lymphocytes, they act indirectly on these cells by producing the antibodies. Followed, they destroy the pathogen indirectly by producing the antibodies, whereas T lymphocyte directly acts against the antigen. Followed, they secrete B lymphocyte, they produce a specific type of antibodies, whereas T lymphocyte, they secrete certain cytotoxins and other chemicals to destroy the pathogen. And B lymphocytes are having shorter lifespan, whereas the T lymphocytes are having they are having a longer lifespan. Followed. So this is all about the T lymphocytes as well as the B lymphocyte. Any doubt? Yes. Don't have one kidney, blood purification affects, madam? No, no, no. No, it will be carried out. One kidney is also sufficient for purification of the blood. As the immunity which is brought about by B lymphocyte is called as humoral immune system, whereas T lymphocyte is called as cell mediated immunity. Is any doubt? Shall I switch on to next part? So these immunities are of two types. The first one is called as innate immunity, which is non-specific, consisting of 4 barriers: physical barrier, physiological barrier, cellular barriers, and cytokine barriers. Second type of immunity is called as acquired immunity, which is very specific based on antigen and antibody reaction. And these antigen, that is antibodies that are present in our body, is made up of 4 polypeptide chains which are arranged in the form of Y-shaped structure. Followed, 2 are called as light chains and 2 are called as heavy chains, represented as H2 and L2. And these chains are held together by disulfide bonds. Each chain consists of 2 regions, variable region and constant region. The variable region consists of antigen binding sites, where the constant region remains the same in all the antibodies. Followed, and the different types of antibodies are IgA, IgD, IgM, IgG, IgE, isn't it? And this specific immunity, that is acquired immunity, is due to presence of 2 lymphocytes. One are called as B lymphocytes, and the immunity brought about by this B lymphocyte is called as humoral immune response. And the immunity or antibody immune response, whereas the immunity brought about by the T lymphocyte is called as cell mediated immunity. So let us switch on to next part called as active and passive immunity. Active and passive immunity. So what do you mean by this active immunity and what do you mean by passive immunity? Now it is clear to you about the antigens and antibodies. Antigens are the foreign substances, followed, foreign substances which induces our immune system for the production of antibodies. So when some antigen, apart from this physical barrier, physiological barrier, cellular barrier, cytokine barrier, when this pathogen escapes and enters our body, what are being produced in our bodies? Antibodies are being produced. Specific type of antibodies are being produced in response to antigens. ಒಂದು antigen ಇತ್ತು ಅಂತಂದ್ರೆ 10 antibodies produce ಆಗುತ್ತೆ. Such type of immunity is called as active immunity. Is called as active immunity. Second type of immunity is called as passive immunity. Passive immunity iddaga, that is when the antigen enters the body of an individual, the antibodies are not at all being produced. Antibodies are not at all being produced. Followed, such type of immunity is called as passive immunity. ಅದಕ್ಕೆ ಏನಂತ ಕರೀತಾರ? Passive immunity. Natural ಆಗಿ, ನೈಸರ್ಗಿಕವಾಗಿ ನಮ್ರೊಳಗ ಆಂಟಿಬಾಡೀಸ್ ಪ್ರೊಡ್ಯೂಸ್ ಆಗ್ತಾ ಇದ್ವು ಅಂತಂದ್ರೆ ಅದಕ್ಕೆ ಏನಂತೀವಿ? ಆಕ್ಟಿವ್ ಇಮ್ಯುನಿಟಿ. ಒಬ್ರೊಬ್ರೊಳಗ ಇಮ್ಯೂನ್ ಸಿಸ್ಟಮ್ ಕಡಿಮೆ ಅದ ಅಂತ ಹೇಳ್ತಿರ್ತಾರ. ಯಾರಿಗೆ ಇಮ್ಯುನಿಟಿ ಕಡಿಮೆ ಅದ ಅವ್ರೊಳಗ ಕೋವಿಡ್ ಆಗಬಹುದು ಅಂತ ಹೇಳ್ತಾ ಇರ್ತಾರೆ. ಹೌದಾ? ಯಾಕಂತಂದ್ರ ಅವ್ರೊಳಗೆ ಏನ್ ಪ್ರೊಡ್ಯೂಸ್ ಆಗ್ತಾ ಇರೋದಿಲ್ಲ, ಆಂಟಿಬಾಡೀಸ್ ಪ್ರೊಡ್ಯೂಸೇ ಆಗೋದಿಲ್ಲ. ಆ ಆಂಟಿಬಾಡೀಸ್ ಪ್ರೊಡ್ಯೂಸ್ ಆಗ್ಲಿಲ್ಲ ಅಂತಂದ್ರೆ, ಆಂಟಿಜನ್ಸ್ ಡಿಸ್ಟ್ರಾಯ್ ಆಗ್ಲಿಲ್ಲ ಅಂತಂದ್ರ ದೇ ಸ್ಟಾರ್ಟ್ ಟು ಡಿಸ್ಟ್ರಾಯ್ ಅವರ್ ಬಾಡಿ ಸೆಲ್ಸ್. ಆಯ್ತಾ? ಸಚ್ ಟೈಪ್ ಆಫ್ ಇಮ್ಯುನಿಟಿ ಇಸ್ ಕಾಲ್ಡ್ ಆಸ್ ಪ್ಯಾಸಿವ್ ಇಮ್ಯುನಿಟಿ. ಆ ಪ್ಯಾಸಿವ್ ಇಮ್ಯುನಿಟಿ ನಲ್ಲಿ ಏನಾಗ್ತಾ ಇರ್ತದೆ ಅಂತಂದ್ರೆ, 1 minute ಐ ವಿಲ್ ರೀಡ್ ಔಟ್ ದಿಸ್ ಒನ್. When a host is exposed to antigen which may be in the form of living or dead microbes or other proteins, antibodies are produced in the host body. Such type of immunity is called as active immunity. Active immunity is slow and takes time to give its full effective response. Injecting the microbes deliberately during immunization or infectious antigens gaining access into the body during natural infection induce active immunity. When ready-made antibodies are directly given to the body to protect against the foreign antigens, it is called as passive immunity. The yellowish fluid colostrum secreted by the mother during the initial days of lactation has abundant antibodies such as IgA. ಆ ಫಸ್ಟ್ ಪ್ರೊಡ್ಯೂಸ್ ಮಿಲ್ಕಿಗೆ ಏನಂತ ಕರಿತೀವಿ, ಕೊಲೆಸ್ಟ್ರಮ್ ಅಂತ ಹೇಳ್ತೀವಿ. ಗಿಣ್ಣದ ಹಾಲು ಇರ್ತದಲ್ಲ, ಆ ಗಿಣ್ಣದ ಹಾಲಿಗೆ ಏನಂತೀವಿ, ಕೊಲೆಸ್ಟ್ರಮ್. Or first produce milk in the body of human beings is also called as colostrum only, and it consists of rich amount of IgA antibodies. IgA antibodies ಹಾಲೊಳಗಿರುವಂತ ಆ antibodies ಇರುವಂತ ಹಾಲು ಆ ಮಗು ಕುಡಿಸಿದ್ವಿ ಅಂತಂದ್ರ what happens, the immune system of the that is newly born baby increases. Followed, that colostrum contains IgA to protect the infant. The fetus also receives some of the antibodies from the mother through the placenta during pregnancy. So these are the examples of passive immunity. ಪ್ಯಾಸಿವ್ ಇಮ್ಯುನಿಟಿ ಒಳಗ ಇದ್ದಂತವ್ರಿಗೆ ಜಾಸ್ತಿ ಡಿಸೀಸ್ ಬರ್ಬೋದಲ್ರಿ ಅಂತ ಕೇಳ್ಬೋದು. ಹೌದು, ಅವ್ರಿಗೆ ಏನ್ ಮಾಡ್ತಾರಂತಾರ, ರೆಡಿಮೇಡ್ ಆಂಟಿಬಾಡೀಸ್ ಆರ್ ಬೀಂಗ್ ಇಂಜೆಕ್ಟೆಡ್. ಫಾರ್ ಎಕ್ಸಾಂಪಲ್, ಈಗ ಸ್ನೇಕ್ ಬೈಟ್ ಆಗಿತ್ತು ಅಂತ ತಿಳ್ಕೊಡ್ರಿ. ಆ ಸ್ನೇಕ್ ಬೈಟ್ ಆದಾಗ ಆಂಟಿಜನ್ಸ್ ಪಾಯ್ಸನ್ ಎಂಟರ್ ಆಗ್ತದ. ಆ ಪಾಯ್ಸನ್ ಎಂಟರ್ ಆದಾಗ ಎಂಟಿ ಪಾಯ್ಸನ್ ಅದಕ್ಕೆ ಇಂಜೆಕ್ಟ್ ಮಾಡ್ಬೇಕಾಗ್ತದಲ್ಲ. ಎಂಟಿ ಪಾಯ್ಸನ್ ಇಂಜೆಕ್ಟ್ ಮಾಡ್ಬೇಕಾದ್ರೆ ಇಂಜೆಕ್ಷನ್ ಮಾಡ್ತಾರಲ್ಲ, ಇಂಜೆಕ್ಷನ್ಸ್ ಒಳಗೆ ಏನಿರೋದಿಲ್ಲ, ಆಂಟಿಬಾಡೀಸ್ ಇರ್ತವೆ. So these antibodies are being injected into the body of the patient to that is mobilize these pathogens, or to that is inactivate these pathogens. Followed. So this is the difference between active immunity and passive immunity. So for right from March, April, May, June, that is people are suffering from, people are scared with this COVID virus. We all the people, we are healthier because of this active immunity only. ನಮಗೂ ಕೋವಿಡ್ ವೈರಸ್ ಇನ್ಫೆಕ್ಟ್ ಆಗಿ ಹೋಗಿರ್ಬೋದು, ಬಟ್ ನಮ್ಮ ಇಮ್ಯೂನ್ ಸಿಸ್ಟಮ್ ಏನದ ಅಂತಂದ್ರ ಆಕ್ಟಿವ್ ಇರೋದ್ರಿಂದ ನಮ್ರೊಳಗೆ ಏನ್ ಪ್ರೊಡ್ಯೂಸ್ ಆಗ್ತಾ ಇರ್ತಾವ, ಆಂಟಿಬಾಡೀಸ್ ಆರ್ ಬಿಂಗ್ ಪ್ರೊಡ್ಯೂಸ್ ಅಗೇನ್ಸ್ಟ್ ದಿಸ್ ಆಂಟಿಜನ್. ಆ ಆಂಟಿಬಾಡೀಸ್ ಪ್ರೊಡ್ಯೂಸ್ ಆಗೋದ್ರಿಂದ ನಮ್ರೊಳಗೆ ಏನಾಗಿಲ್ಲ ಅಂತಂದ್ರ ಅದ್ರ ಇನ್ಫೆಕ್ಷನ್ ಆಗಿದ್ರೂ ಕೂಡ ನಮ್ಗೆ ಅದ್ರದ್ದು ಏನು ಇಫೆಕ್ಟ್ ಆಗೇ ಇಲ್ಲ. ನಾವೆಲ್ಲ ಹೆಲ್ದಿ ಇದ್ದೀವಿ. Because we are having which type of immunity? We are having active immunity. ಬಹಳಷ್ಟು ಜನಕ್ಕೆ ಬಂದದ್ ಯಾಕಂತಂದ್ರ they are having passive immunity, wherein their body fails to produce the antibodies. Followed. So let us study about this vaccination and immunization in the next class, I think. Followed. It is already time. So let us study about this vaccination and immunization. ಇವತ್ತು ಹೇಳಿದ್ದೆಲ್ಲ ತಿಳಿತಲ್ಲ? Types of immunity nalli, one is innate immunity, second one is acquired immunity. Yes, innate immunity is present right from birth of an individual. After birth bandanta immunity ಏನಂತ ಕರಿತೀವಿ, acquired immunity. And this acquired immunity is due to presence of B lymphocytes and T lymphocytes. You have studied about difference between B and T lymphocytes, as well as the differences between active immunity and passive immunity. ಇದರ ಎಲ್ಲ ನೋಟ್ಸ್ ಹಾಕಿರ್ತೀನಿ, ಆ ನೋಟ್ಸ್ ಕರೆಕ್ಟ್ ಆಗಿ ನೋಟ್ ಡೌನ್ ಮಾಡ್ಕೊಡ್ರಿ. ಪ್ಲಸ್ ಥರ್ಡ್ ಪ್ರಾಕ್ಟಿಕಲ್ ಹಾಕಿರ್ತೀನಿ. ಸೆಕೆಂಡ್ ಪ್ರಾಕ್ಟಿಕಲ್ ಬೇಕಂದ್ರು ಯಾರೋ ಸೆಕೆಂಡ್ ಮತ್ತೆ ಥರ್ಡ್ ಪ್ರಾಕ್ಟಿಕಲ್ ಹಾಕಿರ್ತೀನಿ, ಆ ಪ್ರಾಕ್ಟಿಕಲ್ ನೋಟ್ ಡೌನ್ ಮಾಡ್ಕೊಡ್ರಿ. ಥರ್ಡ್ ಪ್ರಾಕ್ಟಿಕಲ್ ನಲ್ಲಿ ದಟ್ ಈಸ್ ಪೋಲನ್ ಗ್ರೋತ್ ಆನ್ ದಿ ಸ್ಟಿಗ್ಮ್ಯಾಟಿಕ್ ರೀಜನ್ ಅದ. ಓಕೆ, ಪೋಲನ್ ಗ್ರೋತ್ ಅದ.
ಮ್ಯಾಮ್, what is the name of second practical, ma'am?
Second practical, it is pollen germination.
Third, ma'am.
Pollen germination.
Third one.
Third one, pollen tube growth on stigmatic region.
Okay, ma'am.
Okay, third practical ಕಳಿಸ್ತೀನಿ ಬರ್ತಾ. Okay. ಆ ಥರ್ಡ್ ಪ್ರಾಕ್ಟಿಕಲ್ ನೋಟ್ ಡೌನ್ ಮಾಡ್ಕೊಡ್ರಿ ಮತ್ತೆ ಒಂದು ಡೈಗ್ರಾಮ್ ಅದ ಡೈಗ್ರಾಮು ತಕೊಂಡಿಟ್ಕೊಡ್ರಿ. Okay. So let us meet on Saturday at 8 o'clock. And meanwhile, I'll send you the notes and you note down all these notes in your notebook. Okay. Okay. Thank you.
Thank you, ma'am.